We are talking about movement into and through membranes. Now, last time when we left off, we were learning these three words: isotonic, hypotonic, hypertonic. And by the way, this is in your book on page, anyone know? 204. 204. Let's see some short video footage on these words.
During osmosis, water diffuses across the cell's selectively permeable plasma membrane. Regulating the flow of water through the plasma membrane is an important factor in maintaining homeostasis of the cell. Let's look at three examples. In an isotonic solution, the concentration of dissolved substances in the solution is the same as the concentration of dissolved substances inside the cell. Likewise, the concentration of water in the solution outside the cell is the same as the concentration of water inside the cell.
Okay, what would happen to the cell if you waited for a while? Which is what we're going to do today in our lab.
The water would
take it in.
Would the cell change size?
Yeah.
No, it wouldn't change size.
Wouldn't change size.
The diffusion of water into and out of a cell occurs at an equal rate. The cell retains its normal shape and pressure. In a hypotonic solution, the concentration of dissolved substances is higher inside the cell than the concentration of dissolved substances outside the cell. Water passes through the cell's plasma membrane into the cell, causing the cell to swell. In plant cells, the plasma membrane swells and presses against the fairly rigid cell wall, preventing the cell from bursting. In a hypertonic solution, the concentration of dissolved substances is lower inside the cell than the concentration of dissolved substances outside the cell. Water passes through the cell's plasma membrane and exits the cell, causing the cell to shrink. In plant cells, the cell's plasma membrane shrinks and pulls away from the cell wall.
That's what we'll look at on Friday. We'll put some cells in a hypertonic solution, a saltwater solution, and we'll see how they shrink away from the cell wall. Pretty interesting stuff. Now, another way that a membrane moves things is through these carrier proteins. The proteins actually will expend energy and move stuff against their concentration gradient. Move stuff from an area where it's low concentration to an area where it's high concentration. That's opposite of what normally happens. Normally, if you had this situation, the green stuff would be wanting to go that way. But here, in this active transport situation, you're moving green stuff the opposite way. It's like a cell that already has a lot of sugar inside it, sees some more sugar, and wants that too. And it has to expend energy and pull the solutes inside of it. And it takes energy. It takes ATP is the energy molecule we studied. And that energy will help pull things inside against the concentration gradient. It would be like if I filled the room with ping pong balls and I spotted out the door another ping pong ball and I wanted to get that one in. Well, what's going to happen if I just open the door? Ping pong balls are going to go out. That's the way they normally go, from high concentration to low concentration. So what I have to do is I have to expend energy to get my extra ping pong ball in, because I want one more, because I hoard ping pong balls. So I'm going to have to open the door and keep the ping pong balls inside, and then slide the one in, and then shut the door without letting any of the ping pong balls out.
So is this entire room filled with ping pong balls?
Yeah, if it would work, yeah. Wouldn't that be great?
That would be so cool.
You have to wade through the ping pong balls.
You could like.
That would be so cool.
Why don't they have
Let's
them
do it.
anymore? Like the ball pits for cheese? Yeah.
Yeah, like the ball pits. Here's an example of the sodium. Listen, we got to get to the lab. We got to get to the lab. Me, up here. Here's an example of an active transport situation. It's called the sodium-potassium pump. It's a protein that works inside the cell.
Active transport occurs when energy is required to move a substance across a concentration gradient. An important active transport pump is the sodium-potassium ATPase pump. which is found in the plasma membrane of animal cells. At the beginning of the process, 3 sodium ions bind to sites on the transport protein. The pump must move the sodium ions from an area of low concentration inside the cell to an area of higher concentration outside the cell. The movement of sodium ions across the concentration gradient requires energy provided by adenosine triphosphate, or ATP.
This guy's got a good voice.
The energy utilized by the pump allows the protein to change shape. In turn, the ATP is converted to ADP, adenosine diphosphate. As the protein changes shape, the sodium ions are released to the environment outside the cell.
Whoa.
Potassium ions from the outside environment binds to the exposed sites on protein.
Slowly but surely.
Once in place, phosphate is released, allowing the protein to change shape. The potassium ions are released to the environment inside the cell.
Huh.
That's cool.
Sodium potassium pump.
Look at that.
Now, so a lot of proteins pull things in against their concentration gradient. We call that active transport. The last thing the book talks about is endocytosis and exocytosis. That's where big vesicles form and where stuff, Drew, up here please,
Sorry.
where stuff is pulled into a cell. Now I showed you a video about this before, but you can see the plasma membrane there engulfing an object and pulling it in and forming a vesicle or a vacuole around that object, a bubble basically, that the object is contained within. And that can be done pulling stuff into the cell, and that can be done releasing stuff from the cell. As you can see here, this vesicle attaches to the membrane and releases stuff.
Mr. What's that question?
Yes,
When's our test on
Monday.
Monday. Test is Monday, tomorrow is biography, Friday is another lab.
I thought we had the test Friday.
Yeah.
No, test is
Did we
Monday.
do biography with people that we did last time?
No,
told me on Friday. Yeah, you said it.
I must have said it wrong. I must have been looking at the wrong thing. Okay, here we see a situation where a vesicle is being released. I've shown you this video before, so I'm going through it kind of fast. See the vesicle? And it's releasing its contents outside the cell. Or here's another situation where it pulls in the new contents and takes it in. This way right here where it pulls stuff in, that's endocytosis. That's endocytosis. And this way right here where it releases stuff, that's exocytosis.
So it's like a zipper or something.
Yeah, that's the membrane. It's the cell's membrane. What's it called when it pulls stuff in? Endocytosis pulling stuff in, exocytosis releasing. And that uses energy as well. Pulling stuff in and releasing stuff. That if you do it in big vesicles, that requires energy. Okay, so what we're going to do now is we're going to investigate this. What I'm going to do is I'm going to give you a membrane, a semi-permeable membrane, and you're going to put a sugar solution in it. Oh, you can turn that off. Thank you, Willis out.